And so to start off, amphibians are massively declining worldwide, and it's partly due to this amphibian chytrid fungus. And the amphibian chytrid fungus is also referred to as the Trachochytrium dendrophytitis, or I'm going to be referring to it as BD throughout the talk. And it's a fungus that causes the disease Chytridiomycosis in susceptible frog species. And because of this mass decline and it's globally distributed, it's sort of gotten a lot of press both in the scientific literature as well as in popular media. And as we've seen this emerging disease, we sort of have been able to see that it's now globally distributed, and it's encompassed of a bunch of different lineages worldwide. And so on the left, I have sort of a global phylogeny of BD, where you can see the different colors pertain to the different lineages. And the one that we're most interested in is the GPL, or the global panzootic lineage, which is the one that is most virulent and the one that is really causing a lot of these frog declines. And as you can see, it is now globally spread across the world, and it's a rapid spread of this disease as we see these frog die-offs. And during the spread of the disease, you could argue that some of the most iconic occurrence and documentations of these spreads have been in the high alpine sierras of California, as well as in the montane rainforests of Panama. So we have well-documented cases of the wave and spread of the GPL-BD based on BD occurrence. And so a little bit of background about these regional waves and spread is in Panama. I'll start with just a map of Panama, and we have to see the spread of BD indicated by the red arrow. And you can see that the start of it was around the late 80s, and then it's sort of progressed across the country anywhere from 17 to 50 kilometers per year. So a really rapid spread of this disease across this landscape. And across these amphibian communities, we see about a 25% prevalence when of occurrence during the spread. And then very similarly, in the Sierras, we see also a spread, a rapid spread of BD. And so on the left, I have a picture or a map of Sequoia Kings National Park, which is a national park in the Sierras of California. And all the dots indicate frog populations, known frog localities, known lake basins. And in white, we have these BD negative sites, so they were surveyed, we didn't find BD. And then the red is where we start seeing BD popping up. So this is 2000s, and then as I progress, we can see that basically we're filling in with the reds and the yellows across these populations, indicating that we have positive BD or BD occurring in these populations. And it's a progression in a wave going from south of the park to the north of the park. And what we find is that in the Sierras, a little bit in contrast to Panama, we have a much slower rate of spread. So it's about 700 meters per year. And the prevalence of BD operates a little bit differently. So you could come to a lake basin, all the frogs, you have no BD prevalence, and then the next season you'll go, and all the frogs will have BD in that one locality. And so we have documented this wave-like spread across these different regional localities, and we know that based on the global phylogeny that it's all the same lineage. But what we really lacked is understanding of BD genetics across these different regional localities. And so we were kind of thinking, we have well-documented cases of this, and we can look across very similar spatial scales to wonder, is the GPL the same in these different localities? And so, kind of just using a viral meme of Spider-Man, we have these two locations that seemingly are looking at each other and sort of perplexed as, are we the same? Or if we remove the mask, could we be different? And so that really was the question that we wanted to ask, was are patterns of genetic variation the same in the Sierras and in Panama? And so how are we going to go about doing this? So we have a really rare and exciting opportunity to leverage the skin swab samples from all those BD occurrence data. And so that was usually used for BD surveillance, so it's presence, absence, and how much BD is there. But we can leverage those to now actually genotype those same archive swab samples. And so that's what we did. We took a pretty even amount of samples from Sierras and Panama across very similar spatial scales, and then we amplified regions of the BD genome, and then we look for variable sites across those different loci. And I'm just going to jump into what we see, and this is a PCA plot of all of our samples separated by Sierras and Panama. So Sierras is in the blue and Panama is in the reddish, and what we find is at this core scale that they're genetically different across very similar scales. So they're definitely different GPL for sure at this scale, but what we wanted to do was take it a little bit, zoom in a little bit on each of these different localities and see what kind of variation or differentiation we can find. And so on the left we have the Sierras PCA, and on the right we have the Panama PCA, and all the colors are just those different sites across the study area. And what we find is that the Sierras is highly structured across the Sierras, and the Panama is sort of a panmixia of all these BD genotypes. And again, this is at the same similar spatial scale. And in fact, across the Sierras, we have patterns of isolation by distance going from north to south across our study area. So the purpley dots and sites are in the northern parts of our study section, and then all the way down to the south are in the green and some of the pinks. And so this was really exciting, and we wanted to just sort of verify and make sure that what we're seeing is similar using a different visualization tool. And so we basically are putting this onto an unrooted tree to see if we find those same patterns. And what we find is that this Sierras tree topology, again, is separating by sites, and we have a strong pattern of isolation by distance. So for example, these are different localities highly clustered on this tree, and then on the other side of the tree we have a lot of the northern localities and sort of spreading across in an isolation by distance pattern to the south. And then if you contrast that with the Panama unrooted tree, we have colors all over the map. It's a panmixia. We don't really have any genetic structure going on here. And so the question that we want to know is, well, what factors might actually influence these different genetic histories? So if we look at these two localities and we think that there might be some differences that drive why we have these histories, the most obvious thing is that they're in different parts of the world. And the Sierras is a very different place than Panama. And that's largely because of these high alpine lakes that are very isolated, and there's these stark, steep, mountainous ridgelines that separate these different lake localities. And then in Panama we have sort of these tropical communities where the topography is much different and not as stark as in the Sierras. In fact, the highest locality that we have is not even the lowest locality in terms of meters in elevation in the Sierras. So that's one major difference. Another one is the amphibian communities, the hosts themselves. And so the Sierra system, we have the mountain yellow-legged frog, which is the only amphibian in a lot of these lake localities, and they're the ones really harboring the brunt of BD and these declines. Versus in Panama, we have a whole host of frog species that have different varying levels of susceptibility, and therefore obviously will impact the transmission rates of BD. And then lastly, because this is a life history section and I'm talking about genetics, a little bit of life history on these animals is that the mountain yellow-legged frog overwinters for about 6 to 7 months of the year. So it's under feet of snow and ice, and it's really not moving at all, and presumably the BD is likely not moving as well until those few months of the year when the frogs emerge. Versus in Panama, we have a tropical climate, and the amphibian communities may not be really relying on a seasonal patterns that we see in the Sierras. And so, going back to our Spider-Man meme, is the same but different, which is really the idea that if when we remove the masses of GPL in these two localities, we find markedly different genetic structure among these same lineages. And what we think is that there might be these environmental and host factors that drive the different demographic histories of this same lineage. And this is sort of where we're going to be taking this project forward to see if we can try to explain it with what we have, these patterns with what we have. And while there's really exciting evolutionary biology questions here, there's some obvious conservation implications. And so these frogs are that are in these communities are declining and are threatened and in conservation of importance. And so in Panama, the BD genotypes might not matter when we start moving frogs in the landscape in the forms of reintroductions or through captive breeding programs. But in the Sierras, these frog populations might have these locally adapted BD genotypes that if we start mixing will lead to unintended consequences. And so there's active translocations and reintroductions going on here. And I think this is a super important point because this is active right now. Frogs are being moved on the landscape right now. And without, I argue, without the context of what's happening with BD across those same conservation strategies, that we're really missing what could be impacting the survival of these species. So with that, I want to just thank a bunch of people from Panama that have done a lot of the field work for this and gathered a lot of the work, and then also in the Sierras and my lab and funding sources. And I'll take any questions.
How different are these different BD lineages in Sierras, and how did they get to be different?
So I think that's sort of the question that we would like to answer with some of the demographic histories, because right now we're just looking at this pattern, and what we would like to know is, does that mean that the introduction histories of it is much different? We assume that it's the same kind of spread, that it was introduced and spread across the Sierras, but the genetics don't really match a story like that right now.
What do you know about the underlying genetic structure of the frog populations themselves? Like, do they show a similar pattern as the BD?
Yeah, so so because it's a little bit more difficult with the Panama data because it's a bunch of different host frog populations, so it's hard to get all the genetics. But for the Sierras, I've also, because we have the swab samples, we can also get frog host genotypes off of that. So we have a similar assay to do the exact same thing, but for frogs. And what we find is structuring and isolation by distance across the Sierras. It's unclear whether it completely matches perfectly. There's some things that are going on in the Sierras PCA that show that's not perfectly matching that isolation by distance across that. Thank you.